I'm thrilled to welcome Dr. Balci. She joined the Department of Pediatrics and the Medical Genetics Program of Southwestern Ontario in 2018. She's a clinical geneticist and clinician researcher with a research focus on rare disease models and gene discovery. Dr. Balci completed her medical degree at Ankara University in the Faculty of Medicine in 2005 and her medical genetics residency training in 2009 from Bascamp University, also in Ankara. She then pursued a postdoctoral fellowship in the Berman Zebrafish Lab at Dalhousie, where she worked on zebrafish models of rare human disease. She completed her Canadian residency training at the University of Ottawa, and she's currently the London site lead for the newly installed Care for Rare Solve project. She's building a research program with a special interest in neurological conditions, focused on exploring new ways to diagnose children and adults with rare genetic conditions, finding new disease genes, interpreting gene variants, and creating animal models in hopes of finding new avenues for personalized treatment. So thank you for being here.
Thank you. Thank you very much for that introduction. Sure.
Unless you really want.
No. No.
Not totally.
I know, but the zebrafish lab is how
All right. Yeah. Okay. Hello. So thank you for inviting me to talk here. I've been to a few seminars here and I really enjoyed them. And today I wanted to, initially the idea was to talk about actually anything to do with chromosome 15. So a lot of the conditions that are associated with that chromosome, we were going to talk about. Oops, should I stand here?
No, well, unless you want your face to be in there, but I can get rid of it.
And talk about Angelman syndrome with Dr. Levine, but I think this is just rescheduled to another day. So I thought what I would do is just give some practical information about some of the online resources that we use every single day in genetics, and sometimes maybe you will use as well, and then go through some of the genetic testing results that you might get, because more and more we're getting some uncertain results or some low penetrance variants that look pathogenic but not so much, and how to interpret those, and end with what else we do in the end. I'm seeing some nods, so that's good.
Yes.
Oh, okay. Oh, you might know it. Oh, you believe you're just being there? There we go. Okay. There we are. Good. Now here's my thing here. Probably is there somewhere? No.
Yeah, on the bottom of the
No. Oh, here, I found it. This? No. This. There we go. All right, I think we're good. Yes, so objectives are to talk about the online resources, talk about some of the microarray results that you particularly, well, really any practitioner may come across, and then talk about some low penetrance variants and next steps after microarray. So just a little bit of intro about what we do in genetics. We deal with rare disorders. They are all individually very rare, but collectively quite common, that affect almost 6% of the population. And most of them affect children. A lot of them cause significant disability. So it is a public health issue nowadays. Canadian Association of Rare Disorders calculated that perhaps 1 in 12 Canadians may be living with a rare disorder, just based on those numbers. And what we do in medical genetics is mostly based on diagnosis. And when the disease is so rare, even having a diagnosis starts the care for that individual. Because when there's no diagnosis, we really cannot give any insight into how specifically treat that individual, manage that family. We cannot give them counseling. And of course, there's no possibility for any sort of treatment if there's no diagnosis. So when we say diagnosis, we always thinking about solving, and that's why I had a puzzle in the first slide. And we think about 50% of all comers that come through the doors of genetics. But this may be an underestimate right now, because we're solving more and more, and some of the genetic conditions are actually solved, meaning diagnosed, before they even show up to the genetics clinic. But I want to say that lately, we do solve about 30% of all of our patients with the new best test that we have called exome sequencing. And again, just a really quick intro into what we do in medical genetics. We have different silos, different subsections, In prenatal, of course, we deal with pregnancies that have any abnormal findings, and we offer genetic testing to them. In metabolics, the team usually deals with all the disorders that are screened from the heel poke at birth, newborn screening. And cancer became so much easier to explain after Angelina Jolie had her ordeal with a variant that increased her risk of having breast and ovarian cancer and went through something. So that's what we do in cancer. We pick out those rare families. And in general, what we all do is see kids and adults with a condition, usually multisystemic, that we think may be genetic. And then just for trivia, since we started off with some celebrities, just want to show up some celebrities and see if you know what they have or who they are, first of all. So this gentleman is obviously Peter Dinklage, and he has achondroplasia. Do you know this little guy from Stranger Things? So he has cleidocranial dysplasia. And this late actor is Vern Troyer, and he had cartilage hair hypoplasia. Now, all of these individuals would have come through genetics at some point through their lives, either to get a diagnosis or get counseling about what their chances of passing it on to their children, whatnot. So in the first part, we'll talk about some of the online resources, talk about some testing in the second part, and what we do later on. So basically, this is the quick links on my browser. What I have what I go to every single day. OMIM is one, big one. Gene reviews is another one that I really want to highlight. And these unique and rare connect are patient resources that have been really useful in our day-to-day practice. Has anyone not used OMIM before? Okay, good. So OMIM is online Mendelian inheritance in men, and is a database of human genes linked to human phenotypes. So it is a huge undertaking to catalog these all together, and they have been doing a great job so far, but really having a hard time to keep up with all the new improvements that have happened in genetics. They consider themselves a knowledge base rather than just a database, because they have so many links to other databases that one can use, and these are not all academic databases, some are patient databases. And just a little bit of history into that. It was founded by Dr. Victor Tugce, who is actually a cardiologist and internist with an interest in genetics, genetic conditions, and is now considered to be father of genetics in many circles. He had a lot of body of work with the Amish population in Pennsylvania, where he discovered a lot of recessively inherited disorders. Then he also worked extensively with the Little People of America, and then identified a lot of dominantly inherited skeletal dysplasias. Then he had this body of work on the phenotypes linked to X chromosome. So he thought, why don't we just put them all together in one catalog? And this was the first little sliver that you see on the left-hand side in 1966. It was initially called Catalogs of Dominant, Recessive, and X-linked Phenotypes, until someone said, let's just call it MIM, Mendelian Inheritance in Men. And then, of course, over the years, with increased interest in genetic disorders and inherited conditions, the volumes grew and grew and grew until this 12th edition. And after that, it was put online, and now it's called Online Mendelian Inheritance in Men. So over the years, the entries into OMIM exploded, really, with the, of course, with the Human Genome Project first, and it just had a really huge increase. And then after that, these are just on the right-hand side, you can see the new entries in OMIM every year. As you can see, there were a couple of blips over the years when genome-wide association studies had their high days in 2007, and then you see their downfall after that. And then next generation sequencing was introduced in 2011, and after that, we identified countless new disorders every year, and we continue to do that. Now, OMIM has two main types of entries. Anyone who's been on OMIM must have been confused at some point as to what they're searching for, because it's not very intuitive, and they don't really show this at the first page, and I think they should. So there are two types of entries. There's genes, human genes, where the entry is all about what the gene is, when it was found, where it was cloned, what does it do, whatever is known about it. And then a phenotype. meaning that it could be a disease, it could be Noonan syndrome, but it could also be a real phenotype, because Dr. McCusick was interested in those, like, oh, Darwinian tubercle of the ear, for example, you can find you can find musical perfect pitch as a phenotype on OMEM. So these are just trivia. Of course, we don't use them anymore. But the main reason why OMEM does what it does is that they also link genes and phenotypes. So what phenotype is caused by which gene variant. And so one frequently asked question, at least I get asked, is what do all those OMEM numbers mean? So for every phenotype or gene, there's a 6-digit number. It's actually quite arbitrary. The ones that starts with a 1 or 2 is just phenotypes found before 1994, so things that have been known for a while. It starts with a 3, it's X-linked. It starts with a 4, it's Y-linked. Starts with a 5, it's mitochondrial inheritance. Starts with a 6, then it's newer, basically, and that's all that matters. But in the scientific literature, you will see that all known genetic disorders will have an OMEM number next to it. So it is an important catalog for us. And so gene entries, as I said, will have actually an asterisk before it. So it will have a little prefix to tell you this is a gene entry. So you're not going to find anything about the disease. And there's about 16,000 of those. So we know we have about 22,000 genes. 16,000 of them are what we call OMEM genes, meaning that we know something about them. Forget about the plus sign, they're getting rid of that. A number sign is a phenotype entry that the molecular basis is known. So for example, Noonan, again, I will bring up, it will have a pound or a number sign or a hashtag sign if you're a millennial. And those we have about 5,000 and then more. If the molecular basis is not known, so it is a phenotype, it is a disorder, but we don't know what gene causes it, it has a percent sign. And if it doesn't have any sign before it, be careful. Those are phenotypes that were described many, many years ago, put into OMEM. Now it's impossible to get rid of them. We don't even know if they're genetic. So even things like lupus, things like some of the vasculitis conditions that we know are not single gene disorders are on there. So if you come across those, we can perhaps ignore. So I just wanted to go through OMEM together just to show some of the things that I talked about and just to go through the actual website. So let's say you have a microarray result in your hand and it says this deletion covers such and such genes, and you just want to know what these genes are. You can just use OMEM for those kind of things. And let's say you find PTPN11 somewhere and you search for it. Yes, we're not. Oh, sorry. Okay, I guess we have to fix it. Yes, there we go. No, that's it's okay. I have all the screenshots. Okay, so we'll just go with this. I am prepared for technical glitches. I think this one. Yeah. Okay. Yeah, we're all good. So I typed in PTPN11 into OMEM. Okay. And then it comes back with a list of things, and you will see that if you click on the one with an asterisk, you'll come to the gene. Here it is. It has an asterisk. It tells it. There's the text is all about the gene. But then here's this very helpful table. This gene causes these known human conditions. And on the right-hand side, there will be some external links that we use day to day. You probably won't need to, but things like ClinVar, Nomad, I literally go and click on every day to see if the variant I found in that gene is known, is pathogenic, is WS, those kind of things. But for the purposes of, in a clinical setting, you put the gene in, you know what conditions it causes. Oh, Noonan, okay. You can click on that little number and you go to Noonan syndrome directly. Now, this is a phenotype entry, disease entry, so, and it has a hashtag sign, meaning that it has solved, it has a gene, a known cause. And now usually we click on this clinical synopsis button. Let's see if I did that too, yes. And then you will have all the features of all the known features of the condition listed. in the same little page. You don't have to click anywhere else, and it goes on a little further. So it's very, very useful to see that. And if you click on phenotypic series, because we know Noonan is not caused just by PTPN11, you will see all the other genes that also cause Noonan syndrome. So for this condition, that's the case. So OMIM, we got to be careful when we're counting how many disorders there are. If you count OMIM, you will see that there are 12 different entries for Noonan. Whereas actually there's only one Noonan syndrome caused by 12 different genes, but that's how it's set up basically. And I'll get back to it in a second. There are some really other useful links on the right-hand side this time that I will suggest you use. There's also something called gene map search, and it's from the first web page where, let's say again, you have a microarray result or just any sort of result that says, oh, this patient has a deletion on chromosome 2p16. And you just want to know what genes there are on there. You can go to gene map search, put in the band, and then you will have a list of all the genes, all the OMIM genes in that region. And you can also click on phenotype only entries, meaning that just show me the ones with the phenotype, and it will just show you the ones with the phenotype, with some other functionalities. What I love is clinical synopsis search. So the clinical synopsis is the list that I showed you earlier. Every phenotype will have this kind of table. So you can just search that. So you can, for example, you have a patient with esophageal atresia in front of you. You put that into clinical synopsis search. And then you will have a list of conditions that have esophageal atresia in their clinical synopsis listed. Now you know that you're not going to click on this first one because it doesn't have a gene. It's not even genetic. We don't know what it is. You could click on Bacterial, but we know that it doesn't have a gene, so you can't really test for it. But you can see what charge is this epidermolysis bullosa, cartilage hair hyperplasia. You can even click on these little boxes on the left and compare selected. Up here, I think, yeah, just like when you're buying a car. And then it will actually compare them side by side for you. You know, right? It's really cool. And then you can have Bacterial there as well. You know you won't be able to diagnose it, but at least you can see it all side by side. This one has microcephaly, this one is brachycephaly, this one has something else. So with the patient in front of you, sometimes it's really, really useful. Okay, and then if anyone has ever tried to read about a condition on OMIM, they will see this really temporal description of what happened with this condition since 1938, which is so not helpful, but you can just go and click on gene reviews right there and go directly to the gene reviews entry for that condition, which brings me to gene reviews. Gene reviews has been available online through PubMed with no restrictions for a long time. We love it. We get most of our information from it. As I said, 5,000 rare conditions. We don't know them all. So we go to gene reviews as much as we can. And you can have now the summary section will give you everything you need really to counsel the patient in front of you, or just have enough information about the condition that you know what you're talking about. And it will be a very quick read. And if you wanted more information, then the rest is there too. So keeping that all in one sort of database is of course a big challenge. And as I said, they're having trouble keeping up. But with that database, just by looking at numbers, we can get so much information. For example, we know that 15% of all known human genes cause a human disease. There's 30% that don't cause a human disease because they're embryonically lethal. If you have a variant in them, you don't even survive. So these are very important genes as well. The rest, we don't know yet. I just think of them as candidate genes. We'll find them soon. And the graph on the right-hand side was just recently updated. These are just numbers of gene discoveries performed with conventional approaches in red and with next-generation approaches in blue. And as you can see, in the last 10 years, we can't keep up. There have been so many. And here's another way of showing it on the left-hand side with exome sequencing. It's the red line. But you see this yellow line here. It's a little bit subtle. This one, and you can see it increased really much in the last 3 to 5 years. These are new gene discoveries that are not yet published. So people have discovered them, but haven't had the time or energy to functionally characterize them yet. So it's just gonna increase, I think, in the coming years. Just a quick look back on gene reviews. It is our new holy book, really. We go there all the time. There's again some useful links on the right-hand side. The summary is again all that you need. There's some resource materials too. So if you don't know what they're talking about here, there's a glossary. And also I just found that out that if you go to MedGen here and click, then it will just pull up all the recent PubMed entries about that condition too. I didn't know that. And then GTR is Genetic Testing Registry. So if you wanted to see, for example, you search for achondroplasia, who tests for FGFR3, you click that and you get all the labs. But we can do that for you too. So. Oops. And there's also some CNVs on there, copy number variants, microarray abnormalities, also on gene reviews, which is great. So 15p12.2 recurrent deletion has its own entry, perfect. They are usually updated quite well. ONEM not so much, but gene reviews very well updated. And really all you need to know to answer a parent's questions will be in the abstract. It will talk about this is inherited dominantly. The majority, 95% of individuals will have inherited the deletion from a parent who may or may not have clinical features, which is one of the kind of lower penetrance CNVs. And then it talks about what you can tell them, what you cannot tell them, and what to do for surveillance and management as well. But really for patients, what we go to is Unique. Unique is a website from the UK developed by patients and families who have had children with copy number variants, microarray abnormalities, for them. So what they do is they have free disorder guides that you can just scroll through all the chromosomes 1, 2, 3, and if you find a deletion or duplication and if it has a guide, you can just print that and give to the family. That's what we have been doing. They are written by families, but they're curated by doctors. Some of them are really good, really helpful, have pictures, but there's of course a little bit of an ascertainment bias. So they're mostly the more kind of severely affected kids on there. And so I always warn the families about that. And then there's a new website called Rare Connect that was European, but now actually housed in Canada, where for all rare disorders, not just microarray abnormalities, patients can go in, find almost like a chat room where other families all over the world have entered in their information, asked a question, and they can just connect with each other there. Social media, basically. Right, so that brings me to genetic testing. Just a couple of, again, entry-level information that I always share with the patients when they come in. Genetic doesn't always mean it's inherited. Always they come and ask, a lot of times they come and ask, well, this has never been seen in my family. Why do I see it in my child? Well, it could have been happening for the first time in your child, or you and your partner may have been carriers of a condition that you didn't know about and it's successive. It could have been X-linked. Mom might be a carrier not knowing about it. Or you're affected too and you don't know it. So there are all these different possibilities for genetic testing. And to explain what chromosomes and genes are, I always use the library analogy. People have used different analogies like cookbook and whatnot. But what I say is that, so in our bodies, we have all these cells. In our cells, we have our genetic material. And all of our genetic material is like our library. So we have 22,000 genes, 22,000 books in our library. Comes in pairs, one from mom, one from dad, so 22,000 pairs. And we keep them on 46 bookcases or chromosomes. Because chromosomes are basically DNA wrapped up around itself, so the pieces of DNA that are compressed. So you can, and on each chromosome are hundreds to thousands of genes, so you can think of them as bookcases. So if you're doing a chromosome study, like a microarray, it's just like walking inside this library with a scanner and scanning the bookcases and seeing if there are like this chunk of books missing or extra. That's a microarray. It won't look at the books, it will just look at
inside the books, it will just look at how many there are. A single gene test would be for achondroplasia, for example. We pick down FGFR3 from the shells, we read through the gene, and we try to see if there's a spelling mistake. So that's a single gene test. And a multi-gene panel will just bring down all the books related to epilepsy, for example, and we'll read through them to see if there's a spelling mistake. Exome sequencing is we'll bring them all of them and we'll read through all of them at the same time because now we have a technology to do that, but we can only really interpret the ones that we know about. So the terminology of genetic testing is quite similar in that sense. If it's a cytogenetic result, you can have a duplication or a deletion. They are also called CNVs, copy number variants. We need to have 2 copies of everything. If there's a missing piece of the deletion, if there's an extra piece of the duplication, so it's a CNV, both of them are CNVs. They can be pathogenic, likely pathogenic, or of uncertain significance, or a VUS, as we like to call them. There's also a different terminology in cytogenetics called ROH, which I'll get to in a second, runs of homozygosity. They can be de novo, meaning that it happened for the first time in the individual, maternally inherited or paternally inherited. So you can see all of these in a report. Molecular genetics has the same kind of terminology. A variant or a spelling mistake can be pathogenic, likely pathogenic, or variant of uncertain significance. It can be heterozygous, meaning that it's only on the 1 copy of the gene. Homozygous, it's on both copies and it's the same change. Compound heterozygous, it's on both copies, different changes. De novo, new, maternal, inherited from mom, paternal, inherited from dad. And just about variants, just a little bit. Is this spelling mistake or copy number variant disease causing? The laboratories usually break them into 5 sections. Pathogenic, disease causing, for sure, it's been seen before, confirmed. Likely pathogenic is just so suspicious, it must be. Likely benign and benign are also very easy to tell. But if you can't put them anywhere, then you'll end up with a VUS.
Yeah.
And I used to think we have them so.
Yes.
And I used to think this was like a 5-way split, but it's more like this, where it's very easy to call the likely pathogenic or pathogenic, or a benign or likely benign. It's the ones that are in between that are difficult to put either way. So microarray, we know it. We just talked about what it is. The gains and losses are called copy number variants, and it's much better than a karyotype. And what you can have are 4 different results, 4 types of results that we can get. It could be negative, meaning that we couldn't find anything. It says normal array report. Could be positive, which is also easy, where it's diagnostic. It has a pathogenic or a likely pathogenic CNV. What you do next is then offer a referral to genetics, of course, and we see them sooner if they have a pathogenic finding. But you can also start with some of the recurrences counseling, and that's what we do as well. Talk about surveillance, link them with support organizations, but we also do parental testing. Sometimes we ask the pediatricians to order that ahead of time. Why do we do that? Well, we want to give them good counseling. Is it going to happen again? And also, is this one of those low penetrance variants? If the parents are negative and it happened for the first time in the child, then great. So it's diagnosed as de novo. There's low recurrence risk. If one parent has the same result, though, now it gets a little more confusing because some of the CNVs on microarrays, so some of the deletions or duplications actually have low penetrance, medium penetrance, meaning that not everyone with the deletion or duplication show the exact same phenotype. So mom or dad with a completely normal intellect and no autism whatsoever may also carry that same condition. So those are usually what we call risk factors to have neurodevelopmental conditions and not the sole reason. And more predisposing factors are needed to show it, almost like a threshold effect. They are more harder to counsel, of course. And it means that one parent carries it. There's a 50% chance with every child they have, their child is going to have it too, but how the child will develop, we don't know. If it's a VUS, that means that it's non-diagnostic. A VUS, we do not give a diagnosis with a VUS ever, unless it's not really a VUS. And we still test the parents to sometimes to just, again, convince ourselves that it's nothing, and sometimes to convince the family as well. You can look at the report for the sample type because sometimes it's different. But if both parents have normal array results and the VUS was new in the kid, now this may be significant, but we just don't know enough yet. What we say to them is come back in 2 to 3 years, we'll look at this again. But if one parent carries it, then we're done. Great. It was nothing. Either it's a low penetrance variant that will, but it's most likely just not pathogenic and negative. We can regard this as negative and look at different tests now. There's a fourth possibility, which doesn't happen all that often, but when it does, it's a little bit exciting. Not so much, but it's just very confusing. So a variant is found that has nothing to do with why the patient was referred. For example, you find a deletion in a breast cancer gene or a colon cancer gene. Now that is a possibility that we used to always bring up when we ordered microarray with everyone. It's good practice to mention again because there's no way to unsee it, and the lab will just report it. Whereas with exome sequencing, when we get down all the books, we actually ask the parents, do you want incidental findings? And they can say no, but for microarray, there's no saying no, so they're just going to get it. It happens like 1% of the time, but and they can refer to us. So I got these slides from Melissa Carter from CHIO, or redrew them because I think this is very helpful in understanding, for example, something like autism spectrum disorder. Is it genetic? Is it not genetic? What is the genetic factor behind it? What we now know that there's a genetic influence on about 60% of autism, which is quite high. But when we look at it from a Mendelian disease point of view, it's only about 10% that we can diagnose with a single gene condition. So there is a big portion, which we think is about 50%, that are associated with what we call common variants. They're inherited. These variants are thousands of them. There's so many. They were found with genome-wide association studies back in the early 2000s. But each of them are associated with very low risks of causing autism. But we think that maybe collectively, if you have a certain combination, then you're more likely to get autism. But there's no way to, so some direct-to-consumer genetic testing companies claim that they can do this, but there's no way to reliably measure this right now. What we can reliably measure are these de novo variants that we can find. For example, a new deletion. Williams, Pataki, Lipsky, Smith, McGinnis, there are known conditions that cause autism, but they're also syndromic. Or some of these genes that we know can cause just autism by itself, non-syndromic or intellectual disability, and they're new in the kids and recurrence risk is low, and so on and so forth. These are easy when we find them. There's these inherited rare variants, the medium penetrance CMEs that we talked about. Some of the genes that we have now found are also like that. A parent may carry it and not show the exact same picture, but they are there. So we think that again, maybe some sort of, maybe for these inherited common variants play a role. For example, a parent who didn't inherit some of the other susceptibility genes, but just the CNV didn't show autism, but the kid did because he got some other variant from mom. Or maybe some environmental effects that we just don't know what they are, which we think also contributes to a big proportion of the causes. So up to 10% in a child with autism, if we have a microarray, we will get a pathogenic result. But pathogenic results are not all equal, especially in autism. There's the non-recurrent CMVs, which are rare. They are high penetrance. If you have it, you have autism. If you don't have it, you don't have autism. These are easy. There's the syndromic ones that we talked about, also easy. There's medium penetrance ones like the 16p11.2, where most parents that carry it have some sort of phenotype, learning disabilities when they grow up. For example, with 16p11.2, there's obesity or being tendency to be overweight. we see mostly if the parent carries it, they are also a little bit overweight. But there are some, especially this one that I'll talk about, 15q11.2, that are almost always inherited, and they have been frequently found in controls. They are very, very low penetrance. I need to talk about 15q11.2 with everybody. So this one is called the VP1, VP2 deletion. Okay, this section is from a blog that has just goes on and on about a rant about this deletion because it has just caused so much confusion in the literature. And it talks about there are a few genetic variants that stare back at us. 15q11.2 is one of these, facing us with the constant question of how we define and perceive genetic risk, not because of its pathogenicity, but because of the confusion that it causes. So here's what it is. That's why I think chromosome 15 can be talked about in like 1 hour, just have a 7-hour of its own. This is the region of the very famous Prader-Willi and Angelman syndrome genes are at. It's called 15q11.2, goes all the way to 13.1. But let me just use this, hopefully everyone can appreciate. So there's BP1 means breakpoint 1, and breakpoint 2 are right here, and breakpoint 3 is all the way down below. Why do we have so much deletions and duplications in these regions? Because these regions, BP1, BP2, and BP3 are practically the same. The sequences are very similar to each other. So when there's a replication, when there's an egg or a sperm being produced, the possibility of an error and a slippage is very high. So if it happens between BP1 and BP2, you might have a deletion of this little region. If it happens between BP1 and BP3, then you have the whole section deleted. If it's on the father's chromosome, you get Prader-Willi. If it's on the mother's chromosome, you get Angelman. That one's easy. But what happens if you just have these 4 deleted? This one causes this one has been associated with neurodevelopmental issues and autism and epilepsy and heart disease for the longest time. But again, it does not include all of these genes that are implicated in Prader-Willi and Angelman. It's just very close to them, but it does not. BP1, BP2 is only these 4 genes that are that have nothing to do with Prader-Willi and Angelman. But because they're just very close to them, they have been wrongly kind of grouped together. There is some of the confusion stems from the actual microarray report. Our own lab says that this CME contains 4 omen genes, these 4 genes, all of which are highly conserved, non-inclusive. This deletion overlaps the proximal end of type 1 deletion found in patients with Prader-Willi Angelman. Why would you put this on the report? You know what happens when they put this on the report? We get all these calls from parents, doctors, everyone. Does my child have Angelman? For some reason, they always ask about Angelman, no one asks about no, they don't, right? Because this overlaps with genes. Yeah, exactly. It's not the same genes, but yes, it overlaps because Prader-Willi usually gets rid of all of them, but it's not really overlapping. We shouldn't really say that. So we're in talks with the lab to get rid of that. Also, Hamilton may say it's a VUS. London will say it's pathogenic. So that's another confusion. Yeah. So here we go. Facts about it though. Okay. Initial studies found that they found this CMV in people with intellectual disability, epilepsy, but this was obviously ascertainment bias because when they then did more and more parental studies and control studies, they also found it in them. So we know now that up to 0.5% of all individuals are a carrier of this CMV. We might have one in this room, right? And finally, in 2010, they did a study and they said the penetrance is 3 to 7%, meaning that if you have this deletion, there's a 3 to 7% chance that you're going to show anything, and a 95% chance that you're not. But it's too late now, and the CMV is almost always classified as likely pathogenic, and this has been dubbed the anchoring heuristic. So it's called anchoring in discussion or philosophy, where an individual depends kind of heavily on the initial piece of information that they have and regard all the other information coming from afterwards in relation to that initial piece of information, which is that's this CMV causes, which it probably doesn't. finally, this is very recent. In August, there was a huge group on behalf of PUC. There's a 15Q1.2 working group that published a study that showed that in people with 15Q1.2, there's only 2% of them have autism. That's even sometimes less than the general population. 5.4% had ID. Okay, now this is a little bit higher. Epilepsy, 3.5%, and congenital heart disease. And they, of course, compared it to the controls and said that this deletion does not increase risk for heart malformations and autism. It does perhaps increase the risk a little bit for ID and epilepsy, but it's only a pathogenic of mild effect size. And since it explains only a small proportion of the phenotypic variability, it is not worth discussing in the developmental clinic or in the prenatal setting, which I think now tells us that we can stop talking about 15Q1.2. What I'm trying to say is that if I have a patient in front of me with intellectual disability and they have this deletion, I will look further. I don't think this explains anything. I will do another test. And has anyone come across something called the runs of homozygosity on a microarray report? Is it also confusing a little bit?
Well, especially because the issue would be, is there consanguinity?
Exactly.
And especially in a lot of the cultures that we see. But then more than that is, are there any deletions or duplications in them?
Right. So usually ROHs are reported like this. I'll just start with the example where they say the microarray result is actually normal, female, but there's AOH. So within those regions, there are no deletions or duplications. The complement is there. The problem is that normally in a partnership, the child will have different combination of both grandparents or sets of grandparents. And if they also marry with someone with outside of the family, their child will also be kind of a mixture. But if there is a cousin marriage, for example, let's say these two are cousins, and the resulting offspring may have similar regions in their genome, whereas if they have unrelated parents, they wouldn't have similar regions on their genome. Another way to explain this is that, let's say this grandma is a carrier of something in this little red box, and that's the black bar, and she passes it, and it's a recessive condition, so she's just a carrier, she doesn't show anything. She passes it on to this son and this daughter. They pass it on respectively to their own children. These cousins get married, and they pass that down, both of them. So now this child may have the same thing on either chromosome, meaning that if there is a recessive gene there and a variant in that gene, they are at risk of having a condition that is recessive. But microarray is actually normal, so there is no CNV, there's no deletions or duplication, but we look at those regions to see if there's a gene that can explain their phenotype. And you can offer a referral to genetics if you have that kind of report. Depending on how much ROH is there, you can actually guess how much they're related. Sometimes we disclose to the parents that they're related and they didn't know that. That happens. And this is such an example. Yeah, it's not. A female with absence of heterozygosity, only about 2%, so they're less than first cousins. So they're actually not that related, but they happen to have that. And here's the clinical information: bilateral megaly, hydrocephalus, and then they will list all of those regions where they look homozygous. They seem to have had the same from each parent. So what we do is we go to a further online resource and then try to see what other genes there are on there that have the recessive inheritance to try to solve. Any questions about that so far, or should we just leave questions to the end? I'm almost finished. So what we do afterwards, let's say a microarray is normal or has a BP1, BP2, and we still haven't solved, we then move on to panels or exome. So panel testing is more specific, related to a phenotype that we see, like autism, intellectual disability, or epilepsy, heart disease, and an exome is more general. There are some advantages and disadvantages of either approach. If we order an autism ID panel, we don't always get the same thing from every company. You might get just 100 and some genes or 1,000 plus genes related to autism and intellectual disability. And if you do exome, of course, now you've opened it up to everything. Right now, autism just by itself, so isolated autism, it does not really qualify for exome. But if there's anything else, then we might be able to order it. There are some best of both worlds kind of approaches that we sometimes use. And again, going back to Melissa's slide, because we haven't been able to do this, the kind of statistics here in London, but from CHIO so far, they've looked at 157 kids with autism and offered the panel, autism ID panel, in 31% of those. And they had a positive result in 7%, negative in 30, and unfortunately 64 of them had a VUS. Just the reality of what we're facing. Exome, well, studies are very different, and a very recent meta-analysis said that the yield is about 36% overall in autism, in neurodevelopmental disorders, so autism, intellectual disability, and everything. But of course, it's a little higher if there are other features, and if it's only isolated, then it's a little bit lower. So even exome doesn't diagnose everything, and which brings me to the little plug that I will have at the end to say, to talk about my research. So what we do is we know that 30 or 40% of our patients are diagnosed with exome sequencing. What about the rest? We know why they're not diagnosed most of the time. There are technical limitations of the test that we're doing, and there are some disease-related issues that are just resistant to exome sequencing. So Care for Rare was the rare disease discovery project of Canada. Oh, that's okay. And started in 2011 as Forge, finding a rare disease genes, and then evolved into Care for Rare. And the outcome is that there were a lot of families sequenced, a lot of disorders solved, and many novel genes identified. And the next step of this project was called Care for Rare Solve, where it Care for Solve has understood that there's this unmet clinical need of all these patients with rare disorders that remain undiagnosed, lots of costs to the healthcare system trying to diagnose them, years of waits for a diagnosis. And it is a pan-Canada effort led from Ottawa and has multiple sites across Canada, and we are one of them. And what we are trying to do are two things. First of all, understand the health system impact of exome sequencing, but also of rare disorders, because it doesn't matter to the ministry when we say, well, Australia did this huge study and this is what they found, and UK did this study and they won't know. We want Ontario data, we want Canadian data. So we're trying to collect that all. It's not that rare disease research hasn't happened in Canada, it just hasn't been in one place. So Care for Solve is trying to get everything in one place, understand the health system impact, convince the ministry to do this test in Canada and not keep sending it to outside the country. And also the next step, of course, is to solve the unsolved 60 or 70%. And to do that, we will be using, of course, new technology, because there are the variants that we know we can pick up with exome. There are the ones that we know that we don't know, and we can pick up with these new technologies that are going to be used. And there are things that we don't know, so we'll see about those as they come up. So I hope I convinced most of you that genetic testing is really, results are really important in the context of the family. And there are some of them, some of the testing results that are not as straightforward as others. But information is really fluid and is building up. And as we move forward, we need to stop anchoring and keep an open mind about what these genetic variants are. Some of them may not be as pathogenic as we thought. But please refer all applicable patients as you see fit, and hope everyone is confident enough to order the first line of testing and interpret them. And with that, thank you very much, and see if you have any questions.
No, just some comments. When I was in after I finished medical school, I discovered that I had lost 2 chromosomes.
Oh.
Yeah, I was taught that before then that there were 48 chromosomes. And then all of a sudden, I
There were
only had
46.
46. I think it was Lejeune, wasn't it, that corrected that error. The other thing that I'm not sure what the current status is for referral for genetic testing. In our day, any youngster that was that entered our doors that was mentally handicapped had biochemical screening. And I think Tony Rupar was one of that group, and he continued that. The other thing I remember, the late Dr. Vizzoni developed a series of slides in terms of various syndromes. And so by looking at an individual like Noonan syndrome, whatever, you could come up with a diagnosis. I'm wondering in these days of facial recognition technology, if it's possible to take a picture of a youngster or an adult and then press a button and say, oh, this is such and such a syndrome.
Yeah.
Is that possible?
It is called face to gene.
Anybody?
It's called face to gene.
Really?
Yeah, there's an app and people are able to download it on their phone, and a lot of pediatricians have started using it. Some geneticists were very resistant and thought that it was, you know, a little bit, what's the word? I can't find the words now.
You have a word
Okay.
finding to do.
I have word finding difficulties because, anyway, threatening to their specialty. I don't believe so. I agree. There is a pattern to be recognized, and we know computers do it better than us sometimes. Not all of the time, but sometimes. And if it helps 1 person out of 10 to get diagnosed a little bit faster, I think it's worth it. So a lot of people have been using it. It's been really good with very dysmorphic syndrome. So Williams, Treacher-Collins is another one that just comes up to my head right now.
Down syndrome?
Oh, yes. So Downs, so right now they're so good with Down that they have even started adding in other ethnicities with Down syndrome, which is more difficult to pick up. So Asians with Down syndrome, Black people with Down syndrome, they're also in that database. It's an initiative of the NIH in the States, and they work with Face to Gene to have as many pictures in it as possible so that there's facial recognition there, for sure. And we used to, I think you would find this interesting, we used to call this forward phenotyping, where you see the patient, you group all of the similar looking ones together, you sequence them all, try to find the gene and establish the disease. Now it's reversed. So we do exome, we look at all of the genes, and we try to find individuals with the same gene and then phenotype them. Oh, by the way, they all have small eyes and lozate ears and short stature, and it goes both ways, really. Because the ones that have been very dysmorphic, very easy to recognize, have all been solved. And now we're coming up to those that are not so recognizable as the ones from before. Biochemical screening still happens, but we are a little bit spoiled by the fact that we can diagnose them all with an exome now. So it's less and less, but don't tell Tony that. he will be upset with me. No, we do that still because it's much faster.
Yeah, so a question I often get in my talks, I'm hearing this all the time, is so why is ASD and ADHD, why is it more common in boys?
Oh.
And so is it because it's X-linked, like a lot of these identified genes?
I can't answer that to perfection, but I do know that yes, there is enrichment of neurodevelopmental genes on the X chromosome for some reason, and it's the only chromosome, of course, that actually maybe there is no enrichment, but the only chromosome that 50% of the population has one less of, and that's probably why, because you know, especially with the and I think again there's anchoring there because a lot of the ID syndromes were first like Fragile X was first ascertained through boys because of this phenomenon. But when we look at all of the neurodevelopmental genes, they're actually all around the genome. maybe that, but it is more common in boys, I guess, or is it? I actually don't know. Is it less diagnosed?
Well, I was
in
gonna say,
girls?
there's also the clinical diagnostic acumen.
Mhm.
In boys, they present with more that leads you to that diagnosis. So in girls, they may be shy, maybe socially withdrawn,
Right.
maybe other issues, you're thinking that as opposed to autism. So there's some of that bias in terms of clinical diagnosis.
Are they underdiagnosed?
Was that-
There?
Yeah,
Yeah, so then that's what the problem is.
Mhm.
And they're often underdiagnosed until they get to the point where their social communication difficulties are causing them functional difficulties.
Right.
And then that's when they come to play. So we see all the little boys as preschoolers,
So
right?
So that's very interesting because with
Yes.
Fragile X, I got a lot of questions lately because we had a prenatal case where we diagnosed, mom was a carrier, so we did the expansion study in the fetus, and it was a girl, and they still wanted to have it done, and it was expanded. And now we don't know what to say, of course, how it's going to turn out because mom also is expanded, but she is functioning.
Yes.
But, we don't know how it's going to kind of close itself with X inactivation and all. But when I look at the literature, it said that girls initially as children look less affected than boys with Fragile X. But when you look at outcomes as adults, they have the same functional difficulties almost as much as boys, which is was very interesting for me to see. Almost as much functional as social anxiety, for example, that they don't function as much as they could, basically.
Yeah, so there's that diagnostic differences in the clinical presentation over time as well.
There must be something genetic.
just one other historical note.
Yeah.
This whole institute was founded by the discovery of Klinefelter syndrome. Dr. Murray Barr,
Oh, of course, yes,
Yes,
Dr. Barr, I knew
yes.
that. Yes,
And
Dr. Barr.
he was the one who said we need to have a facility that can have access to mentally handicapped individuals so that I can find more of our bodies.
There's lots that have been found since then. Yeah, that's true.
That's in the 1960s,
No, that's not your era.
Yeah, I was gonna say, yeah, it was before, come on.
Somebody else is right.
Any other questions?
We talked a little bit about the homozygosity,
Right.
absence of homozygosity. What I've had recently is I've had a number of cases of that, and it depends, and then I always look to see that there's no microarray evidence for deletion or duplication, obviously. And I may or may not know about the degree of consent I'm going to be, because I'm seeing a lot of refugees as well.
Right.
And quite common, okay? But I had a little kid recently who had AOH and was quite high, and the parents were distant cousins. But because of that, it brought up the whole issue about, because there was no deletion or duplication, the whole issue about the recessive gene issue. And this child presented like he was autistic, did not have any significant regression of any kind, but he ended up being Sanfilippo,
End up being.
having Sanfilippo,
Oh.
and doesn't have any of the other findings phenotypically
dear.
at this age. So now we've got a child with Sanfilippo that brings up all the other issues about what's going to happen to this child over time, right? So that was.
So was he diagnosed from those AOHs?
Yeah, so,
Yeah.
well, we had, we were trying to figure out, there were some issues with him, we didn't think, I mean, there was a whole debate whether he had dysmorphic features or not, but they were very mild and subtle, because he looked a lot like family members. So
Right. And
whole
they
issue.
don't, right?
Yeah, that's the
Initially,
point, yeah, they don't. And then we sent him to genetics, and they looked at the AOH, and it was no duplication, no deletion, and they just thought, well, let's see what else. And then came back Sanfilippo. Yeah,
So they
I would
are,
never have guessed.
yeah. And again, you wouldn't have done an exome for him because it's
No.
just autism.
Well, that's what I mean, so.
So that's why microarray actually has a higher hit rate than we initially
Yeah.
think. Not just CNVs, but with AOHs, we actually do diagnose a few very interesting conditions there. Oh, that's sad though.
Yeah. And then, of course, the family now knows, they know quite a bit about it, but he's not showing any symptoms or anything at this point. But of course, the school wants to know what's wrong with him. And the parents don't want us to tell the school because they're still
They
doing
don't?
it. Not at this stage. They're still dealing with all the issues about
Okay.
what does this mean for them, right?
Okay.
And so,
Well, I've had a lot of good experiences with the diagnosis with the school. Not that type of
Not yet.
diagnosis, but when we give a genetic name, a syndrome name to a child, and I write a little letter saying that this is the condition, especially if it's well known and it has a website.
Yes.
Then
yes.
it's just so helpful for the families. I misdiagnosed someone with Cornelia de Lange syndrome. They didn't have the genetic finding, but they looked exactly like it, and they had clinical features all ticked off, and we know that some Cornelia de Lange's don't have
Oh,
genetic.
right. Yeah.
So I wrote a letter, sent the patient back, and also ordered exome just to see if we could find anything else, or find a new gene for Cornelia de Lange or something. They came back and said, thank you so much. It helped us so much. They went online, they looked, and the school was great with it. They implemented all these things. They got her a new chair, blah, 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 which is great. And I had to tell them, actually, now I'm going to take that diagnosis back from you, because it wasn't Cornelia de Lange. It was this new gene called ZD5. And the family immediately asked me, is there a website? And I said, no, it's very rare. And they're like, can you not write another letter? Okay, it's fine. We'll just wait a little bit. Because, you know, when you think about it, it's what the girl has, right? It's her phenotype. It's not her diagnosis. But when we do write that letter to say, I believe this patient has this diagnosis, or we have confirmed a genetic cause, then they just take it more seriously at school.
Well, they take it more seriously than the kid who doesn't have a diagnosis as to why they have neurodevelopmental disorder. That's for sure.
Yeah.
They still believe the neurodevelopmental disorder is related to the environment. And parenting.
And upbringing. And all
Right,
that. Exactly.
right.
Yeah.
Okay, well, thank you
Thank
very
you.
much.
Thank you.